In this video, we will be explaining diffusion and osmosis through experiments. Diffusion is the movement of molecules from a high concentration to a lower concentration. Molecules passively diffuse because as the molecules hit one another randomly, they randomly move to areas of lower concentration where interactions are limited. The diffusion of molecules across cell membranes is passive and involves the movement of nonpolar and small molecules across the cell membrane and into lower concentrations. Osmosis, on the other hand, involves the movement of water from a higher concentration to a lower concentration through a semi-permeable membrane. When there is a higher concentration of a solute in a solution than a cell or other substance, that solution is hypertonic. When a solution is lower in solute, it is hypotonic, and when a solution is equal in solute, it is isotonic. Water is a polar molecule that is attracted to other polar molecules such as sodium or glucose. As a result of this attraction, water will flow to hypertonic solutions where there are more solutes or polar molecules, and from hypotonic solutions until the two solutions become isotonic or equal in solute concentration. This prevents cells from containing too much solute and dehydrating or containing too much water and bursting. Our bodies perform diffusion and osmosis constantly throughout our lives. Passive diffusion allows for the exchange of gases such as oxygen and carbon dioxide across the cell membrane, which allows us to produce the ATP we need to perform activities, breathe, move, and function without giving up more ATP in the process. Osmosis regulates the circulation of water and solutes through our body, allowing us to balance out the foods and drinks we consume, or the activities we perform without worrying about ourselves shriveling with dehydration or bursting with water. These activities, or these processes, are extraordinarily important for maintaining the survival and homeostasis of the cells we're made up of. On top of this, these processes are important to understand when it comes to understanding how the body produces energy and why people struggle with dehydration and overhydration. For our diffusion experiment, we first gathered the materials and set them on the table. We used the sink to fill the transparent measuring cup with half a cup of tap water, and then we placed a single drop of yellow food coloring into the water. We used the timer to measure out 1 minute, and we recorded this process with our phones. Throughout the minute, the molecules in the food coloring had spread evenly throughout the water in the measuring cup. This represents the process of diffusion because it demonstrates how molecules move from areas of high concentration to areas of low concentration. In this case, the drop of yellow food coloring diffused from its original position, which has a high concentration of food coloring in it, into the surrounding water or the area of low concentration, until every part of the water had an equal amount of food coloring in it. This process was passive as well and did not require any energy. For our osmosis experiment, we first gathered the materials. We then used the sink to fill the transparent measuring cup with half a cup of tap water. We then took pictures of the gummy bear before it was used in the experiment, and we placed it into the water. We recorded the process with our phones and measured out 12 hours with a timer. We then pulled the gummy bear out of the water after 12 hours and took pictures of the gummy bear. After 12 hours, the gummy bear was swollen with water and lost a lot of its shape. This is due to the process of osmosis. The gummy bear contained sucrose and glucose syrup, which are polar molecules that water is attracted to. The water surrounding it is a hypotonic solution, as there are fewer polar molecules in the water than in the gummy bear, which is hypertonic. As a result, the water began to flow through the semi-permeable membrane of the gelatin gummy bear in order to try to create an isotonic solution between the gummy bear and the water, which means an equal concentration of solute. This caused the gummy bear to swell. However, the gummy bear did not contain the capacity to hold enough water for the solutions to reach an isotonic state, and it burst, just like a cell will burst if it is placed in a solution that is too hypotonic.